A-Level Biology 9700, Paper 4, Variant 2, February, March 2025, question number 1 and 2. Different types of respiratory substrates can have different energy values and therefore release different quantities of energy when they are respired. Complete table 1.1 to show the energy values of each of the three main types of respiratory substrates. Use one tick to identify which of the two possible energy values is correct for each respiratory substrate. They have given us the table 1.1 and you can see table 1.1 and students, they have given three different types of carbohydrates, lipids and proteins, and the lipids have the highest energy values, and then proteins and carbohydrates are the immediate source of energy, but the energy value is highest in the lipids. So the difference in the energy values of substrates, you can see lipids and then carbohydrates and proteins plus proteins have nearly equal values. Proteins are little bit more than the carbohydrates, but they have given us only two values, so we will go with them. So the difference in the energy values of substrates can be explained. We can explain why the lipids have more energy value than the carbohydrates and proteins. So the energy, especially we can say that the molecular composition is involved in this case. That is specifically how many hydrogen atoms become available when the substrate molecules are broken down. When these molecules, any of them, that is the lipids, carbohydrates, or proteins are broken down, they are broken down, and then ATP is produced or energy is released. So during respiration, hydrogen atoms, play a vital role. So the substrate molecules are broken down and the hydrogen atoms become available. So here I would like to this process, and I want to say that a molecule with a higher hydrogen content, a molecule with higher, a molecule with higher hydrogen content will result a greater proton gradient across to the mitochondrial membrane. So in this way, there will be more ATP. More ATP will be produced and it will show the high energy values. And it happens in lipids because the lipids, they consist of long fatty acid chains. So long hydrocarbon chains, glycerol and fatty acids. So in this way, the energy value is high in them. So I would like to explain that is the more, if there are more hydrogen atoms, then it means more hydrogen transported by NAD and FAD to the mitochondrial matrix, and then the greater, you can say the greater chemiosmosis and then more ATP production. So according to this, we can have that the lipids have 37 approximately, and carbohydrates and protons already told you had nearly equal value. So 17, we will go with the carbohydrates and proteins. Now, next question is part B, determining the respiratory quotient. Determining the respiratory quotient RQ of an organism can be used to indicate the main type of respiratory substrate that is being metabolized in respiration. This is because the different types of respiratory substrates have different RQ values. RQ value means the respiratory quotient. So this is because the different types of respiratory substrates have different RQ values. So table 1.2 shows typical RQ values for carbohydrates, lipids, and proteins. Here you can see that the carbohydrates has RQ value 1, lipid has 0.7, and protein is 0.8. So state the name of laboratory apparatus that can be used to determine the RQ value of organisms such as blowfly larvae. So here I would like to see that there is an apparatus which is called as respirometer. Respirometer consists of two glass tubes in which the oxygen, carbon dioxide, oxygen consumed and carbon dioxide released is measured. So the apparatus name is respirometer. So this name of the apparatus is respirometer, and respirometer is used, uh, you can say, for this purpose, to find out the RQ value. So RQ value is respiratory quotient is equal to carbon dioxide produced and oxygen consumed. So to measure this, the respirometer is used, which is made up of glass tubes, two glass tubes joined together. And the different methods are used to find out the respiratory quotient with this formula. First of all, they will measure the carbon dioxide which is produced and the oxygen which has been consumed, and then they will find out this. Now, the next question is, state the reason for using, when determining RQ values using the laboratory apparatus stated in B1, chemicals such as soda lime or potassium hydroxide solution are used. So state the reason for using chemicals such as soda lime or potassium hydroxide solution when measuring RQ values. So here in this case, when they determine the RQ using a respirometer, chemicals like soda lime CaO, and potassium hydroxide KOH. So these are the two chemicals which are used to find out the RQ value. So these are the two chemicals or solutions which are used to absorb the carbon dioxide produced by the respiring organism. So they are used to absorb the CO2 produced. So by to accurate measure the volume of oxygen consumed, we need to eliminate any change in volume due to the production of carbon dioxide. So to eliminate the carbon dioxide or to absorb the carbon dioxide, or to, you can say, we can measure accurately the oxygen which has been consumed for respiration. So by absorbing the carbon dioxide, any decrease in the gas volume within the respirometer is slowly attributed to the consumption of oxygen. So in this way, to absorb the carbon dioxide produced or to eliminate. to find out accurate measurement of oxygen consumed. Now we are moving towards the next question, and they have given the formula over here for RQ. Organic acids such as malic acid can also act as respiratory substrates when respired aerobically. Their RQ values may be different to the RQ values of the main respiratory substrates. So number of carbon dioxide produced over number of oxygen molecules taken in. So when malic acid is respired aerobically, the equation is C4H6O5 plus oxygen. So 4 CO2 and 3 water plus energy. Calculate how many molecules of oxygen are taken in when one molecule of malic acid is respired aerobically. So when one molecule of malic acid is respired, so we have to find out the molecules of oxygen. So here in this case, you can say you can measure how many oxygen molecules are involved over here. That is there are 5 plus 2, 7. On other side, you can see we have to balance this equation actually. So on this side, you can see 8 molecules of oxygen. There are 3 molecules of oxygen. On this side, 5 plus 2. So it means 7, and there are almost 11. So to balance this equation, on 11 on this side, we need 11 on this side. So we will, if we write 3 over here, then you can have, there will be 6 molecules of oxygen over here. So in this way, there will be a balanced equation. On both sides, the oxygen molecules will be same. That is 11 on this side and 11 on this side. So, and we can say that we need 3 molecules of oxygen. We need 3 molecules of oxygen for the respiration of malic acid. That is 3. We will write over here 3. Now we are moving towards the next question. Now question is calculate the RQ for malic acid. So in the equation, you can see the oxygen molecules are 3. So the formula for RQ, you know, it is carbon dioxide in this equation. You can see the number of carbon dioxide produced and number of oxygen consumed. So number of carbon dioxide produced in this equation, the 4 molecules of carbon dioxide are produced and 3 molecules of oxygen are consumed. So the value is 4.3. If we divide the 4 with the 3, then we will calculate its value. That will be 4 divided by 3 is equal to 1.33. It will be 1.33. So this is the RQ value for malic acid, and we have first of all we have balanced the equation. After balancing the equation, we had find out the RQ value, which is equal to 1.33. That is the 4 molecules of carbon dioxide are produced, 4 molecules of carbon dioxide produced by using 3 molecules of oxygen to break down the malic acid. Now we are moving towards the next part of the question, that is D, the deer mouse. Uh, Pyromyscus maniculatus lives in forests in North America. Shows a deer mouse. The deer mouse is active throughout the year and is much more active during the night than during the day. At certain times of the year, deer mice spend a number of hours during the day in a physiologically controlled state of inactivity, not active. That is called as torpor. This is the specific word used here when the animal or the mouse deer is not deer mouse mice is not active, so that is called as torpor. So during this time, there is a decrease in metabolic rate. There is a decrease in metabolic rate, and you can see graph shows the RQ of a deer mouse from 6 to 22 on a day that included time in torpor. And you can see over here that has the time that has been shown over here from 6 to 22. So moving further, describe the trend shown during torpor in figure 1.3 and suggest an explanation for this trend. So we have to find out the explanation for this. First of all, we will observe the graph and then we will move. So when we look at figure 1.3, so 1.3 is in front of us. The trend which we are observing during the period of that is 10 to 16. So from this point to this point is the torpor time. So during this torpor time, you can see that uh, there is a low respiratory quotient. Respiratory quotient is about 0.9 and little bit increase, and then it is decreasing. And at 18 hours, it is at its lowest point. So till 10 or 14 hours, it is little bit fluctuating, and then it is at lowest position at 18 hours. So, and or here we can see that, so during this period we can from this graph we can come to know that is the respiratory quotient will indicate the primary type of fuels being metabolized by this organism. So here first this organism is metabolizing the it indicating the carbohydrates are the main carbohydrates are the main respiratory substrates. So because you can see over from the uh, here you can see 0.7, 1 and protein is 0.8. So it was in graph 1.2. Now we are talking about this graph, this 1.3. So during this you can see the main uh, which are showing this is at 0. They indicates the then fats are also consuming over here. The fats is consuming because there is the lowest. So RQ values for protein are around 0.8. You can see here we can see there is indication of use of proteins during this graph. So these fluctuations show the RQ values for different substrates. So that is, now I am going to write the answer after this explanation, that is, they are the during torpor, the animal is using all three different types of fuels, that is the carbohydrates, fats, and proteins as well. So the graph indicating that RQ1 at RQ1, the using the carbohydrates and at 0.8 the RQ value indicate the proteins, and at and at 0.7 level, 0.7, the lowest point shows the main respiratory substrate is lipids, because its activity is zero during, it is you can say inactive during this period. So yeah, during this torpor period, the deer mouse enter a state of reduced metabolic due to reduced metabolic activity to conserve energy, is to conserve energy. So the low RQ value, approximately 0.7 during the period shown in figure 1 point, suggests that the deer mouse was primarily metabolizing fats to sustain its reduced metabolic rate. During torpor, this metabolic shift allowed the animal conserve its limited energy reserves over an extended period of inactivity. First of all, so due to conserve energy, then it will primarily obtain energy from carbohydrates when the torpor period is extended. Then it will use reserve energy in the form of lipids. Because firstly, inactive, so it has to conserve its energy. So to conserve its energy, immediately he will, first of all, it will get the energy from the carbohydrates, and at last, when the activity period is long, so to get energy to live live, it will get the energy from the lipids. So all it is, you can say, clear from the graph. 1.3. Now we are moving towards the next question part. Deer mouse have a daily period of torpor only at certain times of the year. Certain times of the year. Focus on over there. Suggest reasons why a deer mouse enters torpor only at certain times of the year. So, the deer mouse have a daily period of torpor only. So, reason is that why deer mouse enters torpor only at certain times of the year. Deer mouse enter as a survival strategy. As a survival strategy for survival. Or in simple words, we can say the deer mouse enters torpor for its survival to get energy due to a change in the environmental conditions. So when the environmental conditions due to less environmental resources. When or in simple words, we can say that when environmental resources are less, so it needs energy. So at that time, for its survival, the deer mice enters in torpor. So here are we can see due to why these environmental resources are decreased, that it has to enter into torpor. So due to low temperature, due to the extreme environmental condition, extreme environmental conditions, I would like to there over due to extreme conditions to maintain body temperature for its survival. Or due to less food resources to conserve energy. One more thing which can be the reason, that is the to conserve the water, to reduce the water loss through respiration and excretion. So it's which can be beneficial in dry conditions that might occur seasonally. So that can also be a reason to reduce water loss. So sometimes it can happen. Next question two. The orca, Orcinus orca, has the largest distribution of all aquatic mammals and is found in nearly all seas and oceans. Or orca are social mammals that usually live in groups, and these groups can vary in size. So figure 2.1 shows an orca. There are a number of distinct types of orca. These distinct types of orca are classified as members of the same species. However, there is evidence that sympatric speciation is occurring. There are two distinct types of orca in the northeast Atlantic Ocean, type 1 and type 2. So type 1 orca feed mainly on fish. Type 1 mainly on fish. Type 2 orca feeds mainly on aquatic mammals such as seals. So figure 2.2 shows the locations in the northeast Atlantic Ocean where type 1 orca and type 2 orca have been observed. Orca do not occur only in these areas, and some groups of orca travel great distance. So you can see the different areas. Orca with the circle has been shown, and the type 2 has been shown with the small here. So you can see the in some areas there is only type 2 orca is available as in these areas you can see, but in this area both of these species are occurring, but in this area only type 1 is occurring, and this area only type 1 is occurring, but there are some where both of these are occurring. So with reference to figure 2.2, explain why the type of speciation that occurring in the orca is described as a sympatric speciation. So, first of all, I would like to tell you that what is happening in these questions. The sympatric speciation is the evolution of new species from a single ancestral species while inhabiting the same geographical region. That is, species are living in the same geographical region, and then you can say evolution, there is evolution. A new species is originated from the single ancestral species. So this is called as sympatric speciation. So here in figure 2.2, I have explained you that the distribution uh, for type 1 and type 2 orcas in the Northeast Atlantic Ocean. So you can see the ocean shows a significant overlap in their geographical regions while the distribution are not identical. You can see there are areas where both types of orcas have been observed within the same general regions of the Northeast Atlantic. So this co-occurrence in the same geographical areas despite the development of distinct ecotypes with different diets is the key characteristics of sympatric speciation. Geographically isolated, it would be allopathic speciation. The fact that they exist in the same broad region suggests that the other factors such as ecological speciation, behavioral speciations can happen. But here you can see already we move towards that is they are also they are you can see they are overlapping here. Both of these species are here, but in the other areas, the difference, these species, they are same species, but they have originated the same geograph. They are living in the same geographical region, but they are now living as the different species. They are overlapping at a place. Both of these species are present. So the fact, the fact that in the same region and that is they are living in the region suggests divergence into separate species. As already told you, that what is sympatric speciation? That is the evolution of new species from a single ancestral species. So here they are present. One more I want to write. They are present in the same geographical area, living as separate species. Now, next question is, suggest examples of behavior separation that would contribute to sympatric speciation of type 1 and type 2. So I think that the contribution to sympatric speciation of type 1 orca and type 2 orca behavior plays a vital role in sympatric speciation by reducing or eliminating gene flow between diverging populations within the same geographical area. So here I would like to say there are some examples of behavior separations. As for example, pre-specific hunting techniques, that is the type 1 orcas feeding on fish would like develop and employ hunting strategies suited for capturing fish. This might involve coordinated movement, specific vocalizations to herd fish and dividing patterns adopted for fish souls. That is the type hunting marine mammals like seals. So both species have developed the different feeding habits. That is type 1 feeding on fish and type 2 hunting marine mammals. So both of these species have developed the different strategies, and in this way, they have brought about the different types of changes in their body pattern and in their behavior to feed on different types of food. And secondly, we can say that the orcas lived in complex social groups, and then difference in prey types could lead to the evolution of distinct social structures uh, and the different feeding. I would like to write over here the difference in prey differences in prey type could lead to the evolution of distinct social structures. And these different social behaviors could lead to, you can say, the assortative matings would lead to uh, where individuals within a given area are more likely to mate with each other with their individuals. So in this way, other thing is, yeah, that is the orcas use a complex vocalization for communication and coordination during hunting. So the type 1 and type 2 orcas have developed distinct dialects or call types. So they also developed the different vocalization methods for communication. So the different, you can see the by applying all these factors, the type 1 and type 2 operas can become reproductively isolated. So both types become reproductively isolated within the same geographical area. Now, next question is, in the Southern Ocean, which surrounds Antarctica, there are three distinct types of orca: type B, type C, and type D. Figure 2.3 shows the locations around Antarctica where type B orca, type C orca, and type D orca have been observed. Type B orca and type C orca are mainly seen near the coastline of Antarctica. Type D orca are mainly seen in the Southern Ocean further away from the coastline of Antarctica. So type B orca, type C orca, you can see the areas. You can see their areas in shore regions of Southern Ocean and offshore regions of Southern Oceans. You can see over these three types. So there are phenotypes differences between the different types of orca. Figure 2.4 shows a diagram of type B orca, type C orca, and a type D orca. You can see the difference in them, the size of all these species. You can see the type B and type D almost, you can say the type B is little bit largest and color pattern is different. Very close, color pattern is very close to the type C, but size of type B is larger than type C, and type D has little bit different color pattern. So with reference figure, state one way in which the type D orca is different from both the type B orcas and the type C orcas. I already told you that type B orca appears to be a moderate size. You can see B is a moderate size, or you can say the larger than both of. A type C is smaller, but they have the same color pattern, color scheme and the same, whereas the type C are depicted as the being smaller in size compared to type B. So we can see it has moderate size. Let me write over here. Moderate size, smaller, but both of they are similar, similar in pattern, color pattern, or you can see. And whereas the D is shown to be distinct in their appearance, you can see the appearance and the color pattern as well. The D, they have a smaller, so you can see this area. You can see over it. So you can see they have much smaller post-orbital white patch. This one, smaller white patch, which is significantly reduced compared to type B and type C. So the overall body shape might also appear slightly different. You can say more slender or more pointed head through this suitable in diagram. So you can see this is little bit more cylindrical in shape. So I'm going to write the answer on the given space. The type D has much smaller post-orbital white patch as to B and C. And their body, overall body shape the overall body shape slightly different slightly different from B and C. You can see this is more cylindrical. Now next question is phenotypic differences between D. Physical appearance. Phenotypic differences are means the physical differences between D or, and the other types of orca shown in figure 2.4 could have resulted from the process of genetic drift, including the founder effect. So suggest how genetic drift could result in phenotypic differences between type D orca and the other types of orca shown in figure 2.4. So phenotypic differences between type D orca and the other types which are shown in figure 2.4 would have resulted from the prior existence of genetic drift in the founding population of type D orcas, including the founder effect. So I think we have to suggest how genetic drift could result in phenotypic differences between type D and the other type of. So genetic drift is the random, first of all, genetic drift is the random fluctuation of allele frequency. The allele frequencies within a population. So or smaller population frequency, smaller population. And the founder effect, this is genetic drift, and the founder effect is what? Founder effect is a specific type of genetic drift. Genetic drift, genetically, they are, you can say, little bit separate, get separated. So that occurs within a new population. So here I would like to write the answer in the lack of interbreeding. So the you can say the lack of interbreeding would allow the genetic differences accumulated through genetic drift in the type T populations to be maintained and potentially amplified over time, so leading to the observed phenotypic divergence. So the different factors are involved in this. First of all, the you can say the small founding populations, and then we can see the different evolutionary uh, points which can occur, and these lack of gene flow. In briefly, I would like to write this answer. The founder effect could have established would have established a unique set of allele frequencies in the ancestral type D orca subsequent genetic drift over time coupled with limited gene flow due to their offshore distribution. And you can see this distribution in the given figure 2.4. Gene flow is decreased when they get separate from each other. So could have further shaped their genetic makeup, leading to the distinct phenotypic characteristics observed in type. Now, the next question. In the future, the different types of orca may be classified as separate species. If so, some of these newly classified species will have very small population sizes. So just two factors other than population size there should be monitored when assessing the conservation status of any newly classified species of orca. And if so some of these newly classified species will have very small population sizes in future. So we have to suggest two factors other than population size that could be monitored when assessing the conservation. So if the different ecotypes of orca, like those in Northeast Atlantic and the Southeast Oceans, are eventually classified as separate species, some of them might indeed have small population sizes, making them more vulnerable to extinction. If these population size is small, they are at the extinct, or you can say at the verge of extinction. So they are the most vulnerable species. So when assessing the conservation status of such newly classified species, it is crucial to monitor factors beyond just the number of individuals. So we can see that we have to monitor their geographical range. So a species with a very restricted geographical range is inherently more vulnerable. So we have to monitor the first of all the first factor is their geographical range. And if the range is small, small range means more vulnerable, more danger for to become extinct. So if their limited habitat is threatened by environmental changes, so climate change, pollution, other activities, the entire population could be at risk, more vulnerable or more risk, you can say. And you can see these, so geographical range should be observed, the geographical range, and it should be extended to save them. The other is their reproductive rates. should be observed and managed to save them from extinction. So their population size increase, population size to increase population size, we can say their geographical area should be increased. They should have to cover the more geographical areas. They should have more habitats. They have the more places to live, and then their reproduction rate should also be increased. And by increasing their reproduction rate or by other factors, we can save them. Uh, we can save them to from extinction. So it was all about question number 1 and 2. The paper downloading was not available. I have used it online. So please like, share, and subscribe my channel to get more papers or more questions of this paper. Thanks for watching.